All right, so today is actually very short. So this chapter obviously was very wordy and explained it very well, but it really just reviews the structure of B cells and T cell receptors, so that's really the only take-home point here. There's not much. Chapter 5 is very, very difficult. I think it's almost 50 pages long. The content is very hard, but next week we're not doing anything, and then the week after we'll cover Chapter 5, so start early to try and get through it. But we'll be done pretty soon today. Okay. So it just starts off with one quick definition that we have heard before. Antigen, of course, are any molecule or part of molecule that is specifically recognized by the T cell receptor or the B cell receptor on the site. Okay, so the first thing we're going to talk about is antibody and the structure of antibody molecules. So, of course, there's 5 classes of antibody: IgA, IgM, IgG, IgE, and IgD. Of course, we know very little about IgD. The structure of all of them is very similar. So it's heavy chains and light chains. Light chains come in 2 different varieties. It could be a lambda or kappa light chain. And in general, there is a ratio of 2 to 1 in humans of kappa to lambda light chains. No one knows why. We can exploit that and actually check that ratio to see if you have an expansion of the antibody. It's a tip-off that you do if that ratio is skewed in one direction or the other. And because everything that is divided, reproduced, whatever it would be, monoclonal is all going to have that same type of light chain. Okay, so on antibody, it's made up of a variable region where the antibody binds and a constant region, and that constant region is the effector function. So the strength of the interaction between a single antigen binding site and its antigen is called the affinity. The avidity is the total strength of all interactions. So an antibody has 2 variable regions, so it can always bind twice. So at minimum, the avidity is twice the affinity for an antibody molecule. Does that make sense? But then on an IgM, which is found in a pentamer, the avidity is actually 10 times the single affinity of one variable region. So an IgG and IgE would be twice. The avidity would be twice. And IgA it would be 4 times because it's created as a dimer. Okay, so here's the basic structure of an antibody. It's roughly a Y shape, so it's 3 equally portioned parts. So it's made up of 2 heavy chains. There are these chains, which are connected by a disulfide bond. And of course, it's connected with 2 light chains. So these could be kappa or lambda, it doesn't matter. And then these have 4 immunoglobulin-like domains. So the variable region is 1 immunoglobulin-like domain, and the constant region are 3. So this is the first, second, third variable region. So you can see that the variable region is actually made up of both the light chain and the heavy chain. This is where antigen binds, And then the constant region is just made up of heavy chain. So the structure of antibody was discovered through several experiments which are partially described. The only takeaway point that I think you need to know from this, it's just kind of tradition that everyone memorizes this Ethane and Pepsin cleavage rules. So if you have an antibody and you put it in presence of Pepsin cleaves it right at the location of the disulfide bond or just above it. So what happens is you get 2 FAB fragments, antibody binding regions, and then 1 FC portion. Pepsin, however, cleaves up in this constant region or parts of this constant region into fragments. So you're really just left with an FAB2 region. So one of my co-fellows years ago, Ethane is in papayas. That's the first thing you'd have to remember for this mnemonic to work. Ethane's in papayas, which are fabulous. The FAB regions are fabulous. And that's how I've always remembered this. Come up with whatever mnemonic you want, but this is actually something that comes up enough, although clinically not a terribly high yield. Okay, so in this antibody binding region, right, this variable region, if you sequence the DNA, you find that there are 3 particular points that are highly variable. So they're represented here by these red spikes. We call these highly variable region 1, 2, and 3. They're also known as complementarity determining regions, or CDR 1, 2, and 3. And the one that is particularly variable is hypervariable region 3, or CDR 3. That is something I would definitely remember. So then once this gets translated into amino acids and then folded, we can actually see that the way antibodies get folded, these end up in the same region. And as it turns out, this is exactly in this antigen binding site right here. So these 3 variable regions are variable for a reason, and so they can bind to different antigens, and they can bind to a wide diversity of antigens. You want them to be highly variable. But this CDR3 is the most variable, but all 3 are very close together. So this book seems to have a lot of these crystal structures of different molecules. I guess not crystal structures, they're just, I don't know, ways of writing how the protein folds. Whenever you see these arrows, that usually represents a beta pleated sheath. It's just a confirmation of folding. And not something you remember, but some of these structures are repeated so frequently in nature that we're pretty good at actually predicting. If you read a DNA sequence and then you translate it into the amino acid sequence, of predicting what that structure is going to look like. And there's software actually that you can go on the government website, the same one that has this PubMed, and there's actually a protein folding program, so you can actually put in an amino acid sequence and it will predict the structure based on the amino acids and their affinity for water and their polarization, and from what we know from experience in nature of what tends to make things like beta pleated sheets. So you'll see a lot of them. I don't think it's something that you really need to hold on to, but I do think this is important, that this concept, this introduction to the importance of the conformation of the protein once it's folded. So in a sequence, these are far apart, but it doesn't matter. Once they're folded, they're close together, and that's going to come up again in just a minute. So okay, so this picture just gives some examples of different kind of conformations in which will bind to different antigens. And again, this is what you want, because antigens are going to come in all flavors. They're going to be different sizes, different shapes, different polarization. So they can actually have a very tight groove, a more wide open groove. They can even have actually a little piece that would poke up and then almost sit inside of whatever it's binding to. So this region, again, is highly variable, and it's even variable in the way it would connect to the antigen, or it binds the antigen. This binding, though, is non-covalent. The antibody, the actual piece of the antigen it's binding to is called the antigenic determinant, or the epitope. And this is where the concept of a conformation comes in again. So you have, I don't think there's a great picture for this, right? few different choices. So if you have a protein, and maybe see these would be the areas that the antibody would recognize. You would only recognize it if it was folded appropriately in, say, these three areas were close together. So this would be a conformational epitope. When it's folded, it can get recognized by antibody. But if this was denatured and became linear like this, antibody would not be able to bind to that particular site again. Not that it wouldn't be able to bind to that antigen, but not that particular epitope. That epitope has been rendered useless. But there are some that are linear continuous epitopes, so the whole antigen binding site in one sequence. And when it's folded, it doesn't matter. It's on the same part. So even when it's denatured, the antibody is still going to recognize it. Conformational epitope and linear epitope. Okay, so very quickly, these are just some examples of the different forces that will reinforce the non-covalent binding of the antigen and the antibody. Again, not something I think is particularly useful information for our purposes, but here, if you'd like to read more about it. So the T-cell receptor is very similar. So it's made of an alpha and a beta chain that roughly represent just 1/3 of an antibody. Alternatively, there are such things as gamma delta T-cells, not covered in this chapter, we'll talk about later. They have a much lower diversity. Unlike antibody, they only have one antigen binding site, not two, and it's never secreted. So B-cell receptor is actually membrane-bound antibody, right? But then eventually, once it turns into a plasma cell, that gets secreted. T-cells do not have anything analogous to that, but similarly has a variable region and a constant region. So this is what it looks like. It's made of an alpha chain and a beta chain. Each have a variable region, each have a constant region. The variable region, similarly, is what binds to the antigen in the same way. And of course, it's going to still have three highly variable regions. The third is still going to be the most diverse. So CDR1, CDR2, and CDR3. CDR3 is going to be the most variable. So it's very similar if you took one FAD region or 1/3 of an antibody molecule. The structure is very similar, but this is always bound to the cell surface and is never secreted. There's not much to point out here. We're going to talk more about this in a later chapter when we talk about cell signaling. But many receptors actually, they have their external region, the transmembrane region, and then a very short cytoplasmic tail, which means it can't do very much. So it has to be in very close proximity to other molecules that will propagate that signal towards the nucleus. But we'll go through those pathways at a later date. Again, just the protein folding structure of a T-cell receptor. So you have your alpha chain, beta chain, the variable regions, and the constant regions. So T-cell receptors are unique in the immune system in that they are MHC restricted, which means that they will only recognize antigen that is presented on MHC. So of course there's two types of MHC. There's MHC type 1, which presents to CD8 positive T cells, and is made of an alpha chain and a beta 2 microglobulin. So we'll read about packaging, and it's a little bit easier to remember this, but MHC type 1 binds 8 to 10 amino acids long, but once you kind of know the whole packaging system, that's kind of an easy fact to remember. This is the structure of MHC. So you have your beta 2 microglobulin and then an alpha chain that actually has three parts, alpha 1, alpha 2, and alpha 3. So it's schematically represented here, alpha 1, alpha 2, alpha 3, in the beta 2 microglobulin. So the alpha chain is actually encoded on MHC, and I don't remember what molecule this is. I'm sorry, I don't remember what chromosome this is. Should. Is that right? Which is so beta 2 microglobulin is on 15, and then the MHC is on 6. So MHC type 2 binds to CD4 positive T cells. It's made of an alpha chain and a beta chain. The beta chain is distinct and different from the beta 2 microglobulin. Both of these chains are encoded on MHC. And this binds to kind of longer, less well-packaged amino acid molecules, more like 13 to 17 amino acids in length. This is a nice picture, very similar to the alpha chain and the beta chain. Each has two parts, alpha 1 and alpha 2, or beta 1 and beta 2. Ends up looking very similar to MHC type 1, but the chains are distinctly different. So if you look at the binding cleft of each, the part that's actually going to hold antigen that's being presented to the T cell receptor, remember it's smaller in MHC1. It's all going to fit nicely in. The ends are going to be closed. But this cleft, the ends are actually open a bit on MHC class 2, so this is going to be longer and it can kind of stick out the sides. But in this empiric text, if you remember, it kind of makes this analogous to a hot dog bun with a hot dog sitting in it. It's kind of an oversized hot dog in MHC type 2. And this T cell receptor is going to recognize parts of both, not only the antigen, but parts of MHC as well. So it has to recognize the combination of both for the interaction to occur correctly. So next to the T cell receptor, like I mentioned, there's the CD4 and CD8. So the T cells have thousands of T cell receptors on them, and either CD4 or CD8 associated with all of them. It's only going to be one of the other. So CD4 and CD8 are known as co-receptors, as opposed to a co-stimulator, something we'll talk about later. But a co-receptor is called a co-receptor because it actually binds to the same thing that the receptor binds to. So the T cell receptor binds to the MHC and the antigen. A co-receptor also has to bind to that same structure. So this is the other kind of factoid that you should take away from this chapter. And remember that CD4 is going to bind to this beta 2 region of class 2 molecule, and CD8 is going to bind to this alpha 3 region. So I've kind of gotten used to drawing these backwards. But I always, the way I kind of remember this is I march it out the same. So I always draw the alpha starting in the same corner. And then, you know, in class 1, it's got 3 segments. So alpha 1, alpha 2, alpha 3. And then whatever's down here is going to bind to the co-receptor. Here, start alpha 1 at the same place. There's just 2 regions straight down, alpha 1, alpha 2, and then beta 1, beta 2. I know it's kind of vague, but that's how I've gotten used to marching it out and remembering where it binds to. It always binds to the same region. So if you start with alpha 1 when you're drawing it, just drawing it in your head in that same part, I think it's a little bit easier to remember. Alpha 3 or beta 2. Oh, that is actually it for this chapter. Make sure I didn't miss any big points. I don't think I did. Talk about gamma delta later. Yeah, I think that's it. So in your mind after this, right, just be familiar with the T cell receptor and B cell receptor structure. MHC, you know, kind of a vague notion of binding. But next chapter is the very long, tough one, and we're going to talk about how those receptors are made. Any questions? All right, I'm going to have a great Thanksgiving.